Welcome, class. Today we're going to go over chapter 12, which is the chapter regarding blood. In this chapter, you're going to describe the primary functions of blood, the characteristics of blood plasma, list the form elements of blood, and identify the most important function of each. Discuss the structure and function of red blood cells and how red blood cell numbers and hemoglobin content may change to produce anemia. And describe ABO and RH blood typing. We'll also discuss the structure and function of white blood cells, explain the steps involved in blood clotting, and define the following medical terms associated with blood: acidosis, serum, hematocrit, anemia, sickle cell, polycythemia, erythroblastosis fetalis, RH factor, leukocytosis, leukopenia, phagocytosis, fibrinogen, and thrombosis. So you need to make sure that you can define all those terms. So blood composition. Blood tissue is formed from plasma, which is the liquid part of the blood. Usually, if you look at a test tube, it's the yellow part on the top. Then we have formed elements. These are different types of cells and cell fragments. Hematopoiesis means the forming of new blood cells. Now, blood plasma is blood minus its cells. Blood plasma is blood minus its cells. The composition is water containing many dissolved substances, such as nutrients. Think of nutrients as our digested food, okay? Salts, hormones, waste products. Also in plasma is going to be proteins such as albumins, globulins, fibrinogen, prothrombin. And you can see a breakdown of all of these in figure 12-1 in your book. Now, the amount of blood varies with size and sex. However, there's about 4 to 6 liters of blood on average in the body, or about 7 to 9% of the body weight. Now let's talk about the formed elements. We have red blood cells, which are also known as erythrocytes. You must know those terms. White blood cells, WBCs, or leukocytes. And of the white blood cells, you have two different types. You have granular leukocytes and agranular leukocytes. Granular leukocytes are neutrophils, eosinophils, and basophils. I always think of Neb. Agranular leukocytes are lymphocytes and monocytes. Then we have platelets or thrombocytes. And the counts are, you'll start to learn these more in nursing school. Red blood cells, 4.2 to 6.2 million per millimeters of blood. White blood cells, 5 to 10,000 on average. Platelets really need to be around 300,000 or higher. And platelets help with clotting, or thrombocytes help with clotting. Now, hematopoiesis, this is the formation of new blood cells. There's two kinds of connective tissue that make blood cells for the body. We have myeloid tissue and lymphoid. Myeloid tissue is the red bone marrow that we've talked about a few times before. All types of blood cells begin in the red bone marrow. Lymphocytes, however, move really, really early on in the stage to the lymphoid tissue and mature there. So the lymphoid tissue is where the lymphocytes move to and mature. So here is that figure 12-1. And if you notice in the middle of the blood here in the tube, the plasma is that yellow top layer, followed by the buffy coat. Buffy coat has platelets and leukocytes in it. And then the red is the formed elements, which is erythrocytes. So plasma is made up again of proteins such as albumins, globulins, fibrinogen, prothrombin, a lot of water in here. And then ions, nutrients, which is basically what happens from digested food, waste products, gases, and other substances. The buffy coat has platelets and leukocytes. Leukocytes are your white blood cells. And remember, we have Neb, which is our neutrophils, eosinophils, and basophils. And then we have our monocytes and lymphocytes. And again, down in the red are your erythrocytes or your red blood cells. Now remember, the term serum is plasma minus clotting factors, which include fibrinogen and prothrombin. So when you hear serum, we're talking about plasma minus the clotting factors of fibrinogen and prothrombin. Plasma contains the clotting factors. So, formed elements in the blood. So, erythrocytes or red blood cells are oxygen and carbon dioxide transporters. Neutrophils is an immune defense by phagocytosis. Phagocytosis is cell eating. Eosinophil defends against parasites. Basophils help in the inflammatory response and in secretion of heparin within the body. B lymphocyte is antibody production. So B lymphocytes, B cells, antibodies. T cells is cellular immune response that destroys virally infected cells and cancer cells. Monocytes, monocytes is immune defenses. It is the largest phagocytes. So when I think of monocytes, I think of Pac-Man cells. They're just going around eating everything. And then platelets are used for blood clotting. So red blood cell structure and function. So red blood cells are disc-shaped without a nuclei, and they appear caved in on both sides. So we call it biconcave. Now the function of red blood cells are to transport oxygen and carbon dioxide because the large biconcave disc shape allows for moving dissolved blood gases. The red blood cell count, we call it a complete blood cell count or a CBC. This is a battery of tests used to measure the amounts or levels of many blood constituents, including white blood cells, CBC with differential will include neutrophils, basophils, eosinophils, just all of them. Now, the hematocrit test is the volume of red blood cells specifically. This medical test in which a centrifuge is used to separate whole blood into formed elements and liquid fraction. Red blood cells are dense, so they're really heavy and they're forced to the bottom of the tube. The buffy coat is white blood cells and platelet fraction, and then the normal red blood cell level is about 45%. Now, the test result can be skewed if someone is really dehydrated. So hematocrit test, just to give you an idea here, once it's spun down in the centrifuge in the right, normal, you can kind of see where the red blood cells, about 45% of the tube. In low hematocrit, it's going to be really low in the tube. It's going to be mostly plasma. And in a high hematocrit, you're going to have more than 50% red blood cells and less plasma. So hemoglobin. Hemoglobin is carried in red blood cells. Anemia is the inability of blood to carry adequate oxygen to tissues caused, for example, by 1, inadequate red blood cell numbers. There's not enough. 2, deficiency of hemoglobin, which is the oxygen carrying part of the red blood cell. Aplastic anemia is unusually low red blood cells caused by destruction in the red bone marrow. So aplastic anemia is usually associated with like leukemias, things like that. Iron deficiency anemia is caused by a deficiency of iron. Sickle cell anemia is a genetic condition that produces an atypical type of hemoglobin. So it's not a normal type of hemoglobin. The hemoglobin, instead of being biconcave in that disc that makes up the red blood cell, in sickle cell, the hemoglobin is sickle shaped, kind of an S shaped or kind of like a hook is what it looks like. And this is a problem because it can get clogged and create problems with blood flow in and out of organs. Pernicious anemia is a deficiency of vitamin B12. And pernicious anemia can happen because maybe the stomach isn't producing intrinsic factor, which is necessary for vitamin B12 to break it down and get it utilized in the body. Or it could just be that someone doesn't have enough B12 in their diet. Now moving on to blood types, we have an antigen. The antigen is a substance that can activate the immune system. So antigen, I think of antigen as like something that's coming in to the body, something that's not a normal part of the body. Antibody is the substance made by the body in response to stimulation of an antigen. So an antigen comes in, our body recognizes, forms an antibody to it. Now, blood types, we call this the ABO system. Type A self-antigens in red blood cells, anti-B antibodies in plasma. Type B is type B self-antigens in red blood cells. It's an anti-A type antibodies in the plasma. And remember, anti is against, right? So type A, it's against B type. Type B is against A type. Type AB have type A and B self-antigens, so there's no anti-A or anti-B, so they're not against A or B. And then type O, there is no type A or type B self-antigens in the red blood cells. Both against A and against B antibodies are in the plasma. Now we go over the ABO system more in future nursing classes, so this is just a general summary. So here, this gives you a good visual of the recipient's blood. So it kind of gives you an idea of, we don't want to see agglutination. Agglutination is when cells clump together. We don't want that to happen. So if somebody has type O blood, they can receive O blood. If they're type A, they can receive O or A. If they're type B, they can receive O or B. If they're type AB, they can receive O or A or B or AB. Now, this is before we start talking about the Rh factor. So if you've ever seen blood or heard of it, you know of it as like A positive, A negative, B positive, B negative. The negative and positive part is the Rh factor. So Rh positive blood means that Rh factor antigen is present in the red blood cells. So that's when you get a positive behind the B or the A or the O. Rh negative gives you the negative sign. That means there's no Rh factor present in red blood cells. No anti-Rh antibodies present naturally in the plasma. Anti-Rh antibodies, however, appear in the plasma of Rh negative persons if Rh positive red blood cells have been introduced. So we need the positives and the negatives to not go against each other here. So this is a breakdown of kind of what you need to know at this point. Universal donor and universal recipient blood. So type O negative is the universal donor. So they can donate to AB, or O. Now, type AB positive is the universal recipient. They can receive blood from any of the others. Now, hemolytic anemia is also called erythroblastosis fetalis, and this can occur only during pregnancy. So, erythroblastosis fetalis is hemolytic anemia, and it happens when an Rh negative mother carries a second fetus. So, it doesn't happen the first time, it happens the second time. The Rh positive fetus, caused by the mother's Rh antibodies reacting with the fetus's Rh positive cells, start to destroy each other. So the baby is positive and the mom is negative. So basically, both the ABO and the Rh systems are often used in combination to identify a person's blood type. And depending on the ABO and the Rh, depends on if you're a universal donor, recipient, or what type of blood you can receive. Moving on into white blood cells. So white blood cells are also known as leukocytes. Their general function is defense, immunity. White blood cell count. So there's a differential white blood cell count reveals proportions of each type of red white blood cell. Leukopenia means abnormally low leukocytes or white blood cells. Leukocytosis is too high, abnormally high leukocytes or white blood cell count. Now, white blood cells, we have granular, which is Neb, neutrophils, eosinophils, and basophils. Neutrophils and monocytes carry out phagocytosis. Eosinophils protect against parasitic irritants and allergies. Basophils produce heparin, which inhibit clotting. And then agranular leukocytes, which are monocytes. These are the largest of the leukocytes. They're very aggressive phagocytes. Remember, they're Pac-Man cells. And then lymphocytes produce antibodies. B cells or B lymphocytes, or they directly attack foreign cells, which are called T lymphocytes. So plasma B cells or B lymphocytes secrete plasma proteins called antibodies. So B cells, antibodies. Then we have memory B cells. These are another type of lymphocytes that are dormant until activated by an antigen that it remembers. T cells are lymphocytes, do not secrete antibodies. T cells directly attack the antigen. So they're like the frontline soldiers. They attack bacteria, viruses, cancer cells directly. Platelets play an essential role in blood clotting. And blood clotting. So this is a cascade that you need to know. So basically, in order, this is what happens. So clotting factors released at the injury site produce prothrombin activator. Prothrombin activator and calcium convert prothrombin into thrombin. Thrombin then reacts with fibrinogen and triggers formation of fibrin. This fibrin traps red blood cells to form a clot. Vitamin K stimulates the liver cells to synthesize more prothrombin. So this kind of shows it. You cut yourself and it goes through what I just talked about is shown in this image. So make sure you know that blood clotting cascade. Now, abnormal blood clots. This is a thrombus. This is when a clot stays in place. It has formed and it is a clot that is not healed necessarily, right? It's just staying in place. This is a condition called thrombosis. Because eventually, as it starts to heal, the clot breaks apart, slowly gets dissolved by the vitamin K, goes throughout the body, and we're all good. But when it stays in place, it's called a thrombus, or the condition is called thrombosis. Now, when that clot dislodges and circulates into the bloodstream, we call this an embolus. So the clot is called an embolus, and the condition is called an embolism. Some other terms, make sure that you understand. You can look them up the back of the book. Is acidosis. Acidosis is when the blood pH drops below 7.35. Okay. Below 7.35 is acidosis. Above 7.45 is alkalosis. We've already gone through most of the other terms. Polycythemia is too many red blood cells. Leukemia is too few white blood cells. And we've discussed some of the other terms here. So this concludes our chapter 12 on blood. So go and study, and you will hear from me in the next one.